low cross compatibility, narrow genetic base, polygenic and trait complex, environmental influence and limited screening tools. Dr. Ashok was mentioning in the morning about intergeneric, interspecific crosses, how that is difficult to do in normal thing, how artificial intelligence is going to help us. Look at this. There is a love between these two guys there. We say in breeding, this is, I sometimes make a funny remark on breeding. We say, in breeding, we interfere with the sex lives of the plants. Basically, we don't allow them to do what they naturally do. We say, you cross with this, you do that, and that's how we try to. Imperative is chili is vital to economic nutrition. Everybody talked about it. Breeding must be predictive, proactive, and accelerated. These are the three things which are very, very important. You should be predictive. You must be able to predict the results. You have to be proactive. Do it before it happens. When a thing happens, it is reactive. You have to be proactive and accelerated. That's the thing. Speed. What does AI do? AI integrates data across genome, field, and environment. This is a conceptual framework. You see genomic data there, phenotypic imaging, as I said, predictive breeding, genomic selection, and ultimately our aim is to have resilient cultivars. We talked about resilience, climate resilience and climate smart. Environmental and food safety data and safe produce. These are the important things. How do we accelerate genetic gain? AI improves both accuracy and speed of breeding cycles, predicts breeding value early. Breeding value is something which a genotype has to have to be useful in a breeding program. It helps you to predict the breeding value, automates phenotyping data collection, models complex trait interactions, because otherwise if you have to do straight interactions, you have to spend a lot of time. As I said, it takes a longer time. So to avoid that, we do model complex trait interactions. Automated phenotyping systems accelerate breeding cycles. That's what the Japanese guy was telling in his presentation. Then we have what we call as high throughput phenotyping. And AI helps in high throughput phenotyping from manual to automated field assessment using drones and keep learning. Drones also Dr. Ashok mentioned in the morning. Applications, stress tolerance via thermal imaging, disease lesion quantification. Because based on disease lesion, we assess the susceptibility or otherwise of a particular genotype. Fruit uniformity and color scoring, very important, you know it. Early capsaicinoid prediction. Capsaicin, as you know, is a pungent principle. You want to breed for different levels of pungency, we need to have early capsaicinoid prediction. So this is a pictorial thing, AI-driven phenotyping, as mentioned, combining multispectral imagery, DL-based feature extraction, and real-time trait quantification. Many of these things, I honestly would like to admit, are new to me. These are learnings for me also. Don't ask me, are you doing all this? Is it possible? I am only exposing you or expecting you to understand the possibilities or potential of these technologies. Advanced genomic selection models, traditional versus AI-based models. GBloop, in a while I'll tell you what is GBloop and what is, you already listened to him in the morning, GEBM, I will come to that in a while. These are different genomic models, just be aware of that. So here comes GBloop, that is genomic best linear unbiased prediction. Genomically estimated breeding value. These are the things which will help you to achieve your results and goals and objectives faster. Then deep learning architects, FNNs. So these are all a little bit of complicated things. I will leave the presentation with you. You can go there, look at it and make a detailed analysis of that. So I just mentioned what is haplotype. It is a set of DNA variants along a single chromosome that tend to be inherited together. So then this is a deep learning architectures used in AI-powered genomic selection for predicting complex chili traits, very, very important. This will help you, once you predict the complex traits, then you can know how to use it, or you can make a strategy or a plan to use them. Then comes to the food safety part. Oftentimes, we also heard several speakers mentioning there is a problem of contamination of chili by fungal, bacterial, viral, or whatever. So here, productivity and safety both are to be ensured. Two pillars are predictive modeling for pest and disease management, Another is hyperspectral imaging for contaminant detection. Because there is aflatoxin contamination which has to be detected. As somebody was mentioning, it is non-destructive method. Predictive modeling in action. What does it do? Uses weather, soil and pathogen data, generates pest risk maps, reduces chemical inputs up to 40%. That's great. Enables early interventions to prevent yield loss. Then AI-enabled predictive modeling. It integrates weather, soil and just a pictorial thing of what I said in the previous slide. Then real-time contaminant detection. This is HSI. So as I said, the contamination because of the aflatoxin or whatever. So this is what it uses, fingerprints of aflatoxins using NIR spectrum. The NIR is near infrared. AI models classify contaminated pots. Pneumatic systems instantly remove contaminated pots. So this is how it works, what I explained there. This is a pictorial representation of that. So there is illumination source and whatever is rejected bin that goes into that, whichever is a contaminated fruit. Sequential workflow of AI and HSI contaminant detection, radiometric correction, feature extraction, classification and ejection. So I mentioned there, those things you can read yourself. Integrated AI breeding pipeline, this is how it looks. Integrated AI breeding pipeline connecting genetic gain acceleration with food safety assurance. That is the theme of this conference. That's why I put together this flow chart or flow diagram. Then combines, again, just to summarize, it combines HTP, GS and HSI-based safety systems, create a closed-loop breeding ecosystem, accelerates improvement and ensures food safety. Holistic breeding ecosystem combining multiple AI-powered technologies. Key takeaways are speed, resilience and safety. Then we also talked about policy. Dr. Dalwai was mentioning about policy recommendations. Invest in national scale AIPB infrastructure, that is artificial intelligence precision breeding. Develop AI genomics, data integration platforms. Ensure India's leadership in chili research and safe exports. Thank you. That's it. 12 minutes. 14.
Thank you so much. Being very precise, just in your time, also timing up we have the audit. He has already told that he will leave this presentation here. Anybody wants for further details, you can get it copied.
Yes.
One or two quick questions are there, one or two, so that he is also got time concerns. So we are also doing the same thing. So quick answer questions, one or two.
I don't think anybody will have questions. Even if you ask, I'll say I don't know. Refer to my. You have a question? Go ahead. Mike, please take the mic. Don't ask a tough question, ask a simple one.
Check, hello.
Mike, kodi avrige.
Sir, thanks for nice presentation. This is nowadays passion towards AI. Really, we need realistic data, accuracy in the data. Because AI is everybody working, realistic data is very important than just dragging things and putting things. Sir.
I am getting the echo. I can't hear you. I think Tikku can explain that. Yeah, please.
Because I think if you all heard Dr. Kang's lecture, what he said is actually happening. He didn't talk nonsense. He is actually doing it and I have seen it. And from 2014, the kind of genomics work that group in Seoul National University has done, that's why we requested him to come. Please go through his and interact with him. You will learn how they have converted 4,800 accessions were first DNA profiled and then they went into core selection. The core selection was then further analyzed and then they found the association of the traits with the sequences. And they are now using actually sequence breeding. So it's not in the air. We need to spend time. Instead of becoming a seed company, please work on the breeding aspects and come out with new traits. Sorry.
Thank you. Thank you, Dr. Tikku.
Impartially. Sometimes human beings will be, they might be partial.
Bias.
Unbiased
it
selection.
removes the bias. I think I mentioned in one of the slides, it removes the bias. which we may have as breeders. Yes? Yeah, a lot of data, yeah. As Dr. Dalwai was mentioning, it is deep learning, machine learning, IoT, everything gets combined. So that's why I didn't want to repeat any of those.
Sir, one last question.
Yeah. Yeah. Sharad.
Sir, aflatoxin is something caused by aspergillus, as I understand. Am I audible, sir?
You are audible, but I can't, there is clear, echo is there, echo.
Aflatoxin you were talking about.
Aflatoxin, okay,
Yes,
okay.
yes. It is caused by aspergillus.
Aspergillus flavus.
Yes. Then
Yes,
why
that's
it has
right.
to be sorted out at the breeding level? Is it genetic? Is it carried forward to the next generation?
Yeah, what happens is certain genotypes are resistant to aflatoxin and certain genotypes are susceptible. So here, by clearly selecting or discarding those which are susceptible, you are selecting lines which have a better resistance to aflatoxin. Yeah? Does that answer your question? Thank you.
Thank you, sir.